Hello, I am Nelly Sélem from EvoDeepMed Lab, and today I am going to present our research about how a pangenome analysis of Clavibacter genus suggests the origin of pathogenicity in tomato bacterial canker. Clavibacter is an actinobacterial genus whose species colonize plants such as grass, wheat, pepper, lucerne, corn, potato, and tomato. In particular, C. michiganensis causes tomato bacterial canker, and it is responsible for millionaire losses at the greenhouses. After years of studying C. michiganensis, we have observed that some of the strains show phenotypical differences. We studied some of this variation by measuring antibiotic profiles, kinetic constant, and plant virulence to 8 representative strains isolated from Mexican sites. Each selected strain contains most of the genes of its clade. We also explore genomic variation, such as plasmid presence-absence from each genome, and finally, to better understand the evolutive origin of C. michiganensis, we carried on a core pangenome analysis. A phylogenetic reconstruction with core genome gene families suggests that C. michiganensis is a grass phytonosis, because it is closer to strains isolated from grass than to any other species of the genus. We conclude that grass may be an intermediary host and that a phytonosis may have occurred. After core analysis, we perform a full pangenomic analysis to find specific genes related to this ecological transition. Here we can see a full pangenome reconstruction of Clavibacter genus, where each circle is a genome and each radius a gene family. We have found gene families in the soft core of Clavibacter michiganensis, that is, gene families that are present in almost all C. michiganensis but underrepresented in other species. Interestingly, some of these families gather in gene clusters, that is, they are physically close in the genome and then may be part of a biosynthetic gene cluster. We discovered 4 gene clusters in the C. michiganensis soft core. Cluster 1 shows an overrepresentation of caseins, while clusters 3 and 4 seem to be metabolite producers. Cluster 2 and 3 are shared with michiganensis strains isolated from grass, while cluster 4 is exclusive from C. michiganensis from tomato. Cluster 3 produces metabolite michiganin A and then serves as a positive control, since it has been largely known that some families of these clusters are gene markers that allow us to differentiate C. michiganensis from other species of the genus. As a last word of caution, we find microbionin, a cluster synthetic to michiganin A that occurs in only one Microbacterium arboreus. This occurrence may be due to horizontal gene transfer. So main message to take home is that bacterial tomato cancer may be a grass phytonosis.